So good evening, everyone. Welcome to the live session in pathology by Dr. Radhika ma'am. She is a MD pathology graduate from CMC Vellore, and she is also a consultant of pathologist in Coimbatore. So here she is with us to discuss clinical based topics of inflammation with MCQs. It will be a really interactive session. So this is useful for your PG, entrance exams, INICET, NEXT, or anywhere else, because we are going to concentrate on the clinical implication of the topic. So today we have our ma'am with us. She is taking in several institutes which are preparing students for PG entrance exams. So this will be really a useful session to you all. Stay tuned. You can just answer in the comment section, as well as you can just give your doubts in the comments so that I'll just ask ma'am to clarify it. Thank you, ma'am, and you can start now, ma'am.
Sure. Thank you for the kind introduction, Dr. Raghu. And I'm very happy to take sessions in medicine. I hope my audio and video is clear. Shall I proceed?
Yes, ma'am, you can proceed now.
Okay, fine. So he has already introduced me. So a little bit about me. So I did my undergraduation from Teni Medical College and post-graduation from Christian Medical College. And I have been the author of a few NEET-PG entrance books since 2018. Okay, so today's session is going to be the pathophysiology of acute inflammation. So I'm not going to discuss the entire inflammation. So today's concentration will be on acute inflammation. So how your acute inflammation is going to apply in your clinical settings, right? So we'll move on to the session now. So I think this video will be useful for second year, third year, final year, and those who are preparing for entrance exams as well. So what I thought is I'll just give a short introduction about the topic. Then we will go to the clinical scenario-based MCQs, right? So first we'll start about what is inflammation. So what is inflammation? That everybody should know. Okay. So medicine is all about observation. So you have to observe a lot of things and learn, right? So you should be very keen in observing your points, right? So that's how you learn from the patients as well. So when you take about inflammation, it's nothing but how your blood vessels and your cells respond to any injurious stimulus. So any injurious stimuli, how your blood vessels and cells are going to respond. That's what is inflammation. So depending upon certain factors, we classify them. So before going into all those classification, so I would like to make clear about certain basics of inflammation. So what will you observe? So how will you identify that a patient has got some inflammation? So all of you can just watch this one. So you have two hands over here. So which do you think it is abnormal? So you have one on the left, one on the right. So which do you think it is abnormal? So you can observe and tell or just guess what are the findings shown over here. Definitely this one is abnormal, right? So what do you observe here? So if you keenly observe, there is some amount of redness. So you are able to see some amount of redness, right? So what is this redness? So this we call it as rubor. So rubor means redness, right? Then if you see, like there is a slight swelling compared to this. This one is normal. So you have some amount of swelling. So this we call it as tumor. Okay. So you have redness, you have swelling. Now, most of you would have gone to your clinicals. So whenever you examine a patient, you start with inspection, palpation, then percussion and auscultation. So whenever you have inflammation, if you're going to palpate any inflamed tissue, usually it will be warm. It will be warm. I'll tell you why those happens. So you will be having some amount of warmth. So that is called calor. Okay. So don't pronounce it as color. It is calor, right? Then if you are going to press that area or if you're going to touch it, the patient will have tenderness. And if you ask, the patient will complain of pain. So this pain, we call it as dolor. Okay. So now, even if you have not known anything about inflammation, just observe. So you always learn by observation. So observation is very important for any medico, right? So even if you have not known what is inflammation, if you're going to look at it, you're going to arrive at what is called the signs of inflammation. So if you now go and take your pathology book and read, they would have mentioned all these as signs of inflammation. Okay. So what are the signs of inflammation? So you have redness, which is called rubor. You have swelling, which is called tumor. You have worm, which is called calor. You will have pain, which is called dolor. So I hope this much is clear. So inflammation is how your body is going to respond to any injurious stimuli. That can be any infection or anything else. Okay, so it can react to any stimulus. So when you have inflammation, you get to see these signs. So you're going to see all these. So all these signs of inflammation was put forward by a person called Celsus. So these are 4 cardinal signs of inflammation. Okay. But later, Virchow added another sign of inflammation, which is called loss of function. So loss of function, that's not going to be permanent. It's going to be temporary, right? So if you have any inflammation, most of us would have underwent at some point of time, we would have experienced how inflammation is. Okay. So you will not be able to move that particular part. You will restrict the movements. So that we call it as loss of function. So this is all about the cardinal signs of inflammation. So I hope it is clear. So if you have any doubts, you can post it in the chat box. Any doubts so far? Okay, so far we have discussed what is inflammation and what are the signs of inflammation. So I'll be dealing with a lot of lateral discussion wherever possible, which are also important. So Virchow is the person who proposed the 5th cardinal sign of inflammation. So having discussed that, we have some other things in our body which goes by the name Virchow's. Any guess? Any other thing in our body which goes by the name Virchow's? In any other part, you can post in the comment if you have anything which strikes your mind. Anything? So you have Virchow-Robin spaces in the brain. You have Virchow-Robin spaces in the brain. And apart from that, you have something called Virchow cell. So Virchow cell, any guess what is this? This is the image of a Virchow cell. So this also called lepra cell or globi. So what is this? So when you see especially lepromatous leprosy, so what happens? You will have sheets of foamy macrophages. You will have a lot of macrophages. So a macrophage will be a large cell with a nucleus. So this is not a H&E stain. H&E stain is a basic stain done in pathology, hematoxylin and eosin. But this is a acid-fast stain, Ziehl-Neelsen staining, right? So what you're seeing here, you might not be able to make out the macrophage properly. But if I draw the outline, I think you will be able to make out something. So all these are macrophages which are having some pink bacilli inside. They are loaded with lot of pink bacilli. They are acid-fast lepra bacilli. So the macrophage which contains a lot of bacilli, we call it as a Virchow cell. So they are usually seen in lepromatous leprosy. Okay. So just because we have discussed about Virchow's, I want to make a mention about these Virchow cell as well. So these are the signs of inflammation. So you have redness, that is called rubor. You have swelling, that is called tumor. You have heat, that is called calor. You have pain, that is called dolor. And you have loss of function. So these are the cardinal signs of inflammation, right? So these are the signs of inflammation. So now, depending upon whether you have your inflammation, which is starting suddenly and when does it end, we have something called acute inflammation and chronic inflammation, right? So what do you mean by acute? So it's self-explanatory. So anything which is for a short duration. So it starts suddenly. So it has got a quick onset and it ends off quickly. So that is what we call it as a acute inflammation. So sudden onset and it will disappear quickly. Whereas chronic inflammation usually starts late. It is sort of delayed, but it lasts long. The duration will be of a little longer period. So this we call it as a chronic inflammation. So anybody can pose any example for acute and chronic inflammation? Anyone? You can be a little interactive. So even if it's wrong, there is no issues. So any example for acute inflammation and chronic inflammation? So most of the inflammation ends with itis. So let us take some example. Suppose your appendix is inflamed. What do you call it as? You call it as acute appendicitis. Suppose your gallbladder is inflamed. You call it as acute cholecystitis. Then if your conjunctiva is inflamed, you call it as conjunctivitis. Like if kidney is inflamed or you have acute inflammation there, you call it pyelonephritis. So you have a lot of itis in your body. But always there is an exception to a rule. So can you tell anything which ends with itis, but it's not predominantly inflammation? It's as some other pathogenesis. Anyone? So most of the inflammation ends with itis and the name of the organ. But if you take osteoarthritis, so don't think it is predominantly inflammation. It's a degenerative joint disease. Just because it ends with itis, don't think it's inflammation. It's a predominantly degenerative disease, but there is some role of inflammatory cells that you should remember. So what you should remember is acute is of quick onset and it goes off quickly, whereas chronic will be for some period and it lasts long. So whatever I have told you, acute appendicitis, if it's for a shorter duration, then acute cholecystitis are all example for acute inflammation. And for chronic inflammation, the typical example which everyone tells you, starting from your second year till your final year and even after that is tuberculosis. So tuberculosis is an example for chronic inflammation, right? So now let me see if anyone is going to make a guess of what part of a body it is. So it's a tubular structure. It has got some attached adipose tissue and the external surface is sort of little congested with these dilated veins. That means it's all acutely inflamed organ. So any guess what is this? Any tubular organ in your body with some attached fat in it? So you can post in the comments anything. So this is an appendix. Because it has got congested, it's a little bit swollen, you have dilated vein. It's not a normal appendix. Obviously, nobody is going to remove a normal appendix. It's an acute appendicitis. You have to go for pathological examination, but it's an appendix. So what you have here is a exudate. So exudate means something which has come out of the blood vessel. It could be a collection of neutrophils and other debris, which is forming a deposit over the surface. So you have some gray-white exudate. So mostly if it's appearing gray-white, it's usually composed of neutrophils. So this is an appendix. Suppose pathology is all about your gross examination and microscopic examination. So maybe final years and others might find it little easy, but for second years, remember, so first, whatever surgery they are doing, they're going to take up the specimen, put it in formalin and send it to the pathology department. Then we make thin slices and then keep it in formalin for a day. Then we see it in the next day. Then we describe how it looks. That's what we call it as a gross examination. Then there is a sequence of process which happens, which we will see in some other video. So it gets processed. Then we get a section. Then we look into the slide. So that's called microscopic examination. Okay. So always pathology is all about clinical correlation, gross and microscopic examination. Suppose if you have seen any acute inflammation, not only appendix. So what is the predominant cell which you get to see? That again is very, very important. So if you observe here, are you able to see or make a guess what cell is it? So you're able to see cells with some lobes. So these are nothing but predominantly neutrophils which you are seeing here. So remember, neutrophils are the cells of acute inflammation. So whenever you have acute inflammation, you will see neutrophils. Whereas in chronic inflammation, you will see lymphocytes. Okay. So you have to know how a lymphocyte appears, how a neutrophil appears, and all those. So this again was a question in INICET exam which happened a few weeks ago. So they have marked a cell and asked what is the cell which is marked. Okay, so this is a normal peripheral blood picture, and what cells are you seeing is all your normal constituents of your blood. So whatever you are seeing in pink is all RBCs. Whatever cells you are seeing with a blue nuclei, they are all WBCs. All these are WBCs. RBCs doesn't have a nucleus, so you should know that. So now you should understand how a neutrophil appears, how other cells appear. So first we'll start with neutrophils. So neutrophil is a granulocyte. It has got some granules in the cytoplasm. Are you able to see the granules? And you see some lobes. They have some 2 to 3 lobes, or sometimes usually less than 5 lobes, which are connected by thin strands. So that cell is a neutrophil. Okay. Next cell, this is a neutrophil because it has got multiple lobes connected by strands. Next, going to this cell, what is it which is striking for you? Maybe for most of the beginners, this cell would appear a little attractive and catchy to your eye. Why? Because it has got a colorful granule, orangish granules. And if you look carefully, they have a spectacle-shaped nuclei. So whatever stains blue is the nucleus. Usually because of the DNA and RNA content, the dyes will make the nucleus stain blue. Okay, the cytoplasm just got orange granules. So if you see something with a spectacle shape and orange granules, it is nothing but a eosinophil. Okay, so first you should understand what is normal. Then only you can understand the pathology, right? Next is this cell. So this cell has got a very clumped chromatin. That means you don't see any lobes or anything. It's just a single cell with a nucleus, which is very neat and very less cytoplasm. So this cell is a lymphocyte. This is a lymphocyte. Next, you see this cell, they have a kidney-shaped nuclei. Look at the nuclei here. They have a little bend or a kidney shape. And the cytoplasm color, you can't directly call it as a blue. It appears a little grayish. There is a grayish tinge to it. So this cell is nothing but a monocyte, right? This is a monocyte. The next, the leaves which we see in peripheral smear, because they are constituted by a very less percentage, you have a cell whose nucleus is completely obscured by blue granules. These are basophils. So what is this? This is not a eosinophil because they don't have orange granules. Can you call it a neutrophil? You can call because it has 2 lobes, but the strand is sort of little big. This is nothing but a precursor of neutrophil, which we call it as a band form, which appears like a band. This is a band form. So band forms we don't see usually in a normal peripheral smear. Whenever your marrow is producing a lot of WBCs, you can see some band forms. But apart from that, all the other 5 cell types, you should be very clear. Okay. So how a neutrophil appears, how a eosinophil appears, how a lymphocyte appears, how a monocyte appears, that must be clear. So why am I telling all these? Because if you're going to see anywhere, even in your heart or in your gallbladder, when you see in your appendix or anywhere, if you're going to see a lot of neutrophils, it means you're dealing with something acute or acute on a chronic inflammation, right? So let me summarize whatever we have discussed so far. So we have discussed about the signs of inflammation. So anyone wanted to tell what are the signs of inflammation? You can post in the comments. So what are the signs of inflammation? So you have rubor, you have tumor, that is swelling, you have worm, that is scalor, you have dolor, which is pain, and loss of function. Loss of function. These are the signs of inflammation. Then we have discussed about acute and chronic inflammation. So acute will start quickly and end quickly, whereas chronic has got a delayed onset and they persist for a longer duration. And we have seen examples for acute inflammation like acute appendicitis, acute cholecystitis. An example for chronic inflammation is your tuberculosis. And most important is we have looked into the morphology of different hematopoietic cells. And the cell of acute inflammation is your lymphocyte, is your neutrophil. And the cell for your chronic inflammation is your lymphocytes. You can have monocytes as well, right? So this is what we have discussed so far. So next, again, you need to observe and tell me certain things which you are seeing here. So this is about the basic things which you need to understand before I'm discussing the pathogenesis of acute inflammation. So with this as a base, let's move on to the pathogenesis. So what are you seeing here? You have a blood vessel, right? You have a blood vessel. So inside this, you have biconcave disc-shaped cells. These are RBCs. Then you have your WBCs. So now observe, how is it flowing? Where are the cells? Where is your plasma? How is it flowing? It's always about observation. So the normal blood flow is called, anyone you would have learned in physiology. How does a normal blood flow? What is it termed as medically? It's called laminar blood flow. So what do you mean by a laminar blood flow? Laminar blood flow means the cells will be in the center, whereas your plasma will be in the periphery. So that's how your blood flows normally. But suppose let us take you have any tissue injury, this flow has to be altered. Then only it can fight against infections. So what are those changes which takes place in the blood vessels and in the cells? So those we call it as the vascular events and the cellular events of inflammation. So certain changes happen in the blood vessel, certain changes happen in these cells, how they go and kill or reach their destination and kill the organisms. So first, let's discuss about what are the vascular events of inflammation, right? So now let us imagine this is a blood vessel. This is a blood vessel. You have the cells here, and this is going to supply a tissue over here. Let us imagine this is some tissue. So you have a infection in this tissue, and there will be some changes which happen in this blood vessel and some changes which happens in these cells as well. So now what happens first in the blood vessel? So you should know the vascular changes. What are the vascular changes which happen? So the first change which happens is there will be a transient vasoconstriction. There will be a transient vasoconstriction. Okay. Then you will have a vasodilatation, right? Then there will be some changes which happens in your vessel wall, which leads to increased permeability. Permeability means normally your blood vessel doesn't allow all the proteins or fluids to go out. So the permeability, the ability to allow certain substances to pass through the walls, that will be altered. So you have stasis. So these are the vascular events which takes place. So why you should know all these is, when you take acute inflammation, most of the time they ask certain things. What is the most characteristic feature? What is the hallmark? So the hallmark for acute inflammation is increased vascular permeability. The vessel wall becomes permeable. And how does your vessel wall become permeable? So I hope all of you know your blood vessel is lined by endothelial cells. Endothelial cells. So you should remember certain things about endothelial cells, which you can apply in neoplasia as well. So each cell has got certain property. So all those you should know. So when you take any endothelial cell, how can I confirm that it's an endothelial cell? So you need some immunohistochemical marker. So you do certain special tests to identify or confirm it. So you have certain markers like CD34, which can stain up these endothelial cells. Like, to make it a little easy, it's like a signature of an endothelial cell. So all of us have an address. Like that, if you want to identify something, each cell will have some CD markers. Like if you take a lymphocyte, if you want to call it as a B lymphocyte, that will have some marker, some CD marker, right? You have CD19 and 20. If you want to call something as a T cell, you have CD3 as a marker, right? So like that, endothelial cell will have CD34 as a marker. So if you have any tumor of the endothelial cell or a blood vessel, like your angiosarcoma, they are positive for CD34. So this one particular point you should remember, that is important. The next thing what you should remember is, do you have anything else in your endothelium? Your endothelium has got some granules. They are called Weibel palette bodies. They are called Weibel palette bodies. So what are these Weibel palette bodies? So they store certain substances. One is 1 Willebrand factor, the other one is P-selectins. We'll be talking about P-selectins later, but remember, your Weevil palate bodies is going to store these 1 Willebrand factor and P-selectin, right? So far what I've told you is what are the vascular changes which happen. So your blood vessel will initially constrict. It will initially constrict. So there will be less blood flow. So you will get blanching, right? Then you will get increased blood flow because of the vasodilatation. So I was telling about the various signs of inflammation. So imagine your blood flow is more. What happens? There will be some amount of redness. And because of the increased blood flow, there will be warmth. So the rubor in the inflammation and the warmth, what do you call warmth as? It's called calor. So both of these are due to vasodilatation. Just because your blood vessel is going to dilate, you will get a lot of blood flow, you will get warmth. So the signs of inflammation are related to the vascular changes also. The most important hallmark. So when anyone tells about the hallmark, means it's like a signature of inflammation. You will have increased vascular permeability. So how your permeability will increase? So your endothelium normally will not have any gaps, but if you want any substance to pass through, there will be endothelial gaps. One of the most important factor in increasing the permeability is formation of endothelial gaps. Okay, your endothelial gaps will be there. So what happens if you have endothelial gaps? Certain substance, your fluids and the cells can come out and they can form something outside. So they can form like a collection. So this is what you get a swelling or the tumor. So whatever I have told about the signs of inflammation, they're all related to all these changes. So imagine once your fluid is coming out, what will happen here? You will have a lot of cells but less of fluid. So this leads to stasis. So these are the vascular events. Okay. So you have vasoconstriction, vasodilatation, increased permeability, and stasis. So the hallmark for acute inflammation is increased vascular permeability, which is due to formation of your endothelial gaps. It's due to endothelial gaps. And all these rubor and warmth is all due to vasodilatation, and your tumor is due to increased permeability. So this again was asked in JIPMER 2 years back. The rubor in inflammation is due to what? It's due to vasodilatation, especially your arteriolar dilatation. Okay. So most of the other changes which happens in inflammation happens in the venules, especially the post-capillary venules. Okay. So far you have to remember all these. Then I've told about the endothelial cells, about endothelial cell. They are positive for CD34 and they have Weevil pellet bodies which has 1 Willebrand factor and P-selectin. Okay. So this is about the vascular events. So far we have seen about the various signs of inflammation, what is acute and chronic inflammation, and about the various vascular events. So now at this point, I would like to discuss an MCQ, which is like an applied question. So a 17-year-old boy presented with pain in the right iliac fossa, rebound tenderness was present, lab investigation showed neutrophilic leukocytosis. Which of the following cell is implicated in the pathogenesis of this condition? So let's split this question. You have a young boy with right iliac fossa pain. So if I add one more point to this, you would be able to diagnose this. So if I tell that there is McBurney's point tenderness, what would be your diagnosis? It's a acute appendicitis. So what you need to understand this question is the WBCs are increased. That's what we call it as leukocytosis. And your neutrophils are increased. That's neutrophilic leukocytosis. So what is the cell implicated in these acute inflammation? So I hope undoubtedly what will be the answer here? Anyone, what will be the answer here? The answer is neutrophils because it is acute inflammation. Okay. So next we are going to move to the next event of inflammation, which we call the cellular events of inflammation. So I told, suppose you have the pathogen over here. You have, this is the bacteria. then you have all your cells inside. Your cells have to come out and eat up and clear your bacteria. So how does your cell come from the blood vessel to the site of injury? So all those events, we call it as the cellular events. So these are the events which happens. One is margination, rolling, adhesion, transmigration, chemotaxis, absonization, phagocytosis, and killing. So this might appear a little complicated, but then if I explain what is margination, rolling, etc., you will find it easy to understand. So just have these terminologies in your mind. As I explain, it will be easier for you. Okay. So roughly to compare it, maybe I have some comparison. So you can have any of your comparisons. So it's like a journey of a student from your hostel to some hotel to have food. So let us think the food is the target or the bacteria, and you're moving from the hostel to the hotel. So you need to have a lot of things happening. You have to come out of your hostel, then you have to travel to your hotel, then your food must be made tasty, then only you will eat it. So the similar thing, the bacteria has to be made tasty and it has to travel all the way from the blood vessel to the site of injury. So that's going to be a journey of your WBCs, right? So that's why from the center of the blood vessel, what do these cells do? They marginate. Margin means coming to the periphery of the blood vessel. This is called margination. So I told normally your cells will be in the center. Now they are going to come to the periphery of the blood vessel. This we call it as a margination, right? So once they have come to the margins, next what will happen? They will roll over these endothelial cells. They will roll over these endothelial cells. So this is an endothelial cell with a nucleus. So what do you mean by rolling? So rolling means transient adhesion. That means for some time they attach to the endothelial cells. So initially you will not be able to firmly adhere. It will transiently roll over these endothelium before they come out. Like you slowly peep out of the hostel to see if someone is watching you. The same thing happens. It will transiently adhere. So how does it adhere? So it's always like a lock and key mechanism. So you have a WBC, maybe a neutrophil. Then you have here your endothelial cell. So you should have something on the WBC, something on the endothelial cell, which is going to fit in like a lock and key. Then only it can come out of the blood vessel. So you have certain proteins which are going to help in this. So remember, rolling is because of a group of proteins called selectins. So selectins are a group of proteins, right? So I've already mentioned about one selectin. Anyone remembers about it? Yes, P-selectin. Where are they present? They are present in the Weber pellet bodies in the endothelium. So now I'll introduce you to two more terminologies. You have E-selectin and L-selectin. So what you should remember is selectins are nothing but certain proteins which are going to help in the rolling, which is a cellular event in acute inflammation. It's going to help in rolling. So what is P-selectin? The name is self-explanatory. That means they are present on the platelets and endothelium. They are present on the platelets and the endothelium. So what do you mean by E-selectin? E-selectin is a protein which is present only in the endothelium. Whereas L-selectin is a protein, or you can compare it to a lock. Okay, it's present in the leukocytes. Okay, they are present in the leukocyte. Now, let me draw a blood vessel and explain you all these. So this is a blood vessel. Here you have your WBC. Here you have your endothelial cell, right? So it's like a lock and key. If you understand this, you will be able to understand the further discussions. So what you should remember is rolling is because of something called selectins. You have P-selectin, E-selectin and L-selectin depending upon their location. So imagine these are locks. So to open, you need a key. So anything which is on the platelet will have their receptor in the some other location. If you have something on the leukocyte, the complementary thing to bind will be present on the endothelium. So it fits in like that. Okay. So what is the receptor for P-selectin? What's the receptor for P-selectin? So that you should know. So the receptor for P-selectin is sialyl-Lewis 10. So it's called sialyl-Lewis 10. So that is present in the, where should it be present? So P-selectin is present in the platelet and the endothelium. Okay, so it's a protein. So let's imagine that's the P-selectin, and let me draw a platelet. Okay, maybe this is a platelet, a small cell. So you have this protein P-selectin on the platelet. You have it on the surface of the platelet and the endothelium. Okay, let me draw this one. Let us consider this is the P-selectin, the gray ones. So it has to go and bind to some receptor. So where is that present? So that must be present on the leukocyte. So it will be complementary. So anything which is present on the platelet and the endothelium will have this complementary binding region in the leukocyte or the WBCs. So let us take L-selectin that is present on the leukocyte. Then where will it go and bind? It has to bind to something on the endothelium. So what is present in the endothelium? You have something called CD34. So that's how these complementary things goes and binds. So you have different selectins, your P-selectin, E-selectin, and L-selectin. So the receptor for P-selectin is sialyl-Lewis 10, which is present on your leukocytes. So similarly, the receptor for E-selectin is also sialyl-Lewis 10, which is present on the leukocyte. The receptor for L-selectin is CD34, which is present on the endothelium. So you have something else which is present in the endothelium that's called glycan. That's nothing but a cell adhesion molecule, or you have something called MADCAM. So other cell adhesion molecules, those are receptors. So what you should remember is anything which has its lock on the platelet will have its key in the leukocyte. Anything which has its lock on the leukocyte will have its key in the endothelium. Okay, so it's like a lock and key mechanism. So the receptor for P-selectin and E-selectin is sialyl-Lewis 10. The receptor for L-selectin is CD34. So why am I telling this? Because when you have defect in this sialyl-Lewis 10, which is a receptor for these selectin, you can have certain diseases. So those diseases are called leukocyte adhesion deficiency. Especially you have selectin deficiency or some absence of this sialyl-Lewis 10, you have something called leukocyte adhesion defect 2. Okay, so that's called leukocyte adhesion deficiency. So because you don't have this protein, your leukocytes cannot adhere, it cannot come out, it cannot kill any organisms. So I hope so far this is clear. So all the mechanisms happen in a similar way. So this is only about rolling, which is a transient process. So what you should remember is rolling is because of selectins. So you have different types of selectin, P-selectin, E-selectin, and L-selectin. The receptor for P-selectin and E-selectin is sialyl-Lewis 10, which is present on the leukocyte. The receptor for L-selectin is CD34 and glycan, which is present on the endothelium. So if you have defect in this protein called sialyl-Lewis 10, you have something called leukocyte adhesion deficiency 2. Okay, and this particular defect can be associated with short stature or sometimes Bombay blood group. So we will have a discussion on blood group later. But why is this related to blood group? Because you have an enzyme defect. That's why you don't have sialyl-Lewis 10. So the enzyme which is defect is fucosyl transferase. So the same enzyme is also involved in certain reactions in formation of these blood group antigens. That's why it's associated with Bombay blood group. So 3 points you have to remember is rolling is because of selectin, the receptors for different selectin, and what happens if you don't have selectins, right? So now let's move on to the next pathway, which is margination, rolling, and the next one will be adhesion. So what do you mean by adhesion? So adhesion means a firm attachment. It tightly attaches to the endothelium. So you have your endothelial cells, you have your WBCs, which are going to firmly attach, tightly attach. So what is the protein which is going to help in adhesion? So remember, adhesion is because of integrins. Rolling is because of selectins. Adhesion is because of integrins. So like I told, your P-selectin, E-selectin, and L-selectin, you have different types of integrins. We call it as beta-1 integrins and beta-2 integrins. Okay, so you have different types of integrins. So likewise, you have your Lewis 10 and CD34 as receptor. Your integrins will also have some receptor. So what is the receptor for beta-1 integrin? So the receptor for beta-1 integrin is called VCAM1. That's called vascular cell adhesion molecule. Vascular cell adhesion molecule. Whereas the receptor for beta-2 integrin is called ICAM. Okay, that is also a cell adhesion molecule. That's called intercellular cell adhesion molecule. Both are present on your endothelial cells. Okay, so this is about your integrins. Sorry. Okay, so you have different types of integrins. So you have beta-1 and beta-2 integrins. So the receptor for beta-1 integrin is VCAM. The receptor for beta-2 integrin is ICAM, and both of these are present on endothelial cells. So let's imagine your integrins are not present. What will happen? Your WBCs will not go and adhere, so it can't come out of your blood vessel. It can't kill any organism. So here we have an interesting thing which is happening when you don't have integrins. So let us imagine the patient doesn't have beta-2 integrins. So there is defect in beta-2 integrins. What happens? So like I told you, have leukocyte adhesion deficiency 2 or LAD2. If you don't have beta-2 integrins, you have something called leukocyte adhesion deficiency 1, which is called LAD1. So LAD1 is due to defect in integrins, especially your beta-2 integrins, beta-2 chain of integrins, right? So we have one interesting clinical feature when you have this defect. What is that? So I told your cell has to come out of your blood vessel. You need all these rolling, your adhesion and everything going on. If you don't have integrins, what will happen? Your WBCs cannot adhere, so they will stay inside the blood vessels. They can't come out. So what happens is you have a condition called delayed separation of umbilical cord. So how many of you have noticed that when your umbilical cord fall after birth? Maybe after a week it has to fall. And do you know why your umbilical cord falls after that? Because at the base of your umbilical cord, your umbilical stump, your WBCs will come here. A lot of WBCs will come here. They will release certain enzymes. So they will degrade all those proteins, and that's how your umbilical cord sheds. So now imagine you have WBCs which can't come out because you don't have integrins. What will happen? They will remain inside the blood. They won't release any enzymes, so your umbilical cord cannot separate. So you have something called delayed separation of umbilical cord. So delayed separation of umbilical cord can be seen in leukocyte adhesion deficiency 1. Because they can't adhere, they can't kill any microbes also. So they are prone for recurrent infections. They are prone for recurrent infections. And one more thing is they remain inside the blood vessel. They can't come out. So you can have increased WBCs as well. That's called leukocytosis. So this is about your leukocyte adhesion deficiency, which happens if you have defect in integrins, that is LAD1. So if you don't have integrins, this cord is not going to separate. You will have recurrent infection and you will have leukocytosis. So this is about your margination, rolling, and adhesion. So let me summarize whatever I've told so far about rolling and adhesion. So rolling is a transient adherence of your cells to your blood vessel, whereas adhesion is a firm attachment. Rolling is because of selectins, whereas adhesion is because of integrins. So you have different types of selectins, P-selectin, E-selectin, and L-selectin. You have different types of integrins, beta-1 and beta 2 integrins. So the receptor for P-selectin and E-selectin is silyl-Lewis-10. The receptor for L-selectin is CD34 and glycam-1. The receptor for beta 1 integrin is VCAM1. The receptor for beta 2 integrin is ICAM1. When you have defect in this particular protein or selectins, you have something called leukocyte adhesion deficiency 2, which can be associated with Bombay blood group and short stature. If you have defect in beta 2 integrins, okay, especially a CD18 molecule which is involved in this beta 2 integrins, you have LAD1, which can be associated with delayed separation of umbilical cord. So this must be very clear. So you can easily remember, you write 1 like this, I like this. So leukocyte adhesion deficiency 1 is due to integrin defect, whereas 2 is due to selectin defect. So this is like your margination and rolling. So now you have come out of the hostel, slowly peeping through the wall. Nobody has watched you. So you have rolled and you have here come out of the hostel. Next, this is the most important, is crossing the gate. That we call it as a transmigration. So this is very important MCQ. So you have this process where your WBCs will have a foot-like process and they come out. This particular process of coming out of the blood vessel, we call it as a diapedesis or transmigration. So they usually ask the receptor which is involved in it. So the receptor which is involved is called PCAM1 or CD31. This is called platelet endothelial cell adhesion molecule. That is called PCAM1 or CD31. So remember only one thing about transmigration, it's also called diapedesis, and you have PCAM1 or CD31 involved in this particular process. Now you have come out, you have to go to the hotel. So you need certain attracting factors to go there. So those attracting factors is what we call it as a chemotaxis or the chemotactic factors. So you don't randomly choose some place. You need something which is very attractive, right? So that's what is called chemotaxis. So you have come out of the hostel, you're on your journey to a hotel. So what if somebody is going to call to a library? Are you going to go? No. So it's like a unidirectional. Your thought is everything. I have to go to hotel and eat food. Okay. So remember, chemotaxis is also like that. It's a unidirectional. So once from inside your blood vessel, let us imagine this is your bacteria. Your WBCs, the focus of your WBCs, which have transmigrated, will be towards your bacteria. It will not go either ways, somewhere else and lose its direction. It's always unidirectional. So why is it unidirectional? What is attracting these WBCs towards that particular side? So those agents, we call them as a chemotactic agents. So that you should remember. So you have certain chemicals, certain proteins, which can act as chemotactic agents. So you can have any mnemonic, whatever you want, but what I would suggest is CIL, like your cervical intraepithelial lesion. So that's the mnemonic for chemotactic agents. So what are those chemotactic agents? Your complement 5A, interleukin 8, and leukotriene B4. Your leukotriene B4. Okay. Complement 5A, interleukin 8, and leukotriene B4. So these are produced inside your body in response to your bacteria, which is going to attract your WBCs in a particular direction. So sometimes it could be the bacterial product itself can attract your WBC to that particular site. So far we have discussed about your margination, your rolling, your addition, then transmigration, then chemotaxis. Okay. So I've already summarized what happens in rolling and addition. Now let me summarize what happens in transmigration. So I told you, remember only one thing, it's called diapedesis because you have foot shape processes, and that's because of CD31 or PCAM. Whereas chemotaxis, remember it is unidirectional. And what are these chemotactic agents? It could be either your bacterial products, your complement 5A, interleukin 8, and leukotriene B4. So you have slowly marginated, come out on your way to your hotel. Then you have reached the hotel. The next step, you have sat on the table and you're going to order some food. So next thing is called opsonization. That means making the food tasty. Now, the food for your WBCs is nothing but the injurious stimuli, either the bacteria or any product of cell injury. So that needs to be coated by certain things to make it tasty. That's what we call it as opsonization. So what do you mean by opsonization? So if anyone is going to give you a bread, obviously you will have a very bad face. You're not going to eat it. But suppose if it's made something which is delicious with egg and other stuff, you get interested in consuming it, right? So opsonization is similar process. It's making your bacteria tasty for your WBCs so that they can get attracted and kill them easily. But it doesn't mean always you need some opsonization to happen. WBCs can kill bacteria even without opsonization, but preferentially kills those which are coated with tasty substance. So what are those tasty substance which helps in opsonization? So this again, we have a mnemonic. You can remember CSF. So you have complement 3B, your serum proteins. You have some proteins in your blood, like your fibrinogen, like your C-reactive protein or your MAC-binding lectin protein, then FC fragment of immunoglobulin. So all these will coat your bacteria. Suppose if this is the bacteria, all these opsonins will coat them so that your WBCs can easily go and eat them. So remember, mnemonic is CSF, complement 3B, your serum proteins, and FC fragment of immunoglobulin. Whereas the mnemonic for chemotactic agents is CIL, complement 5A, interleukin 8, and leukotriene B4, right? So this is about opsonization. Now you have something delicious on your table. The final step is phagocytosis and eating it. So that's what we are going to discuss. So how is your phagocytosis going to happen? So phagocytosis means cell. Cyto means cell. Phago means eating. So it is cell eating. So you have other process which we'll discuss later. But phagocytosis means your cells are going to eat up the bacteria. That's called cell eating. So let us consider this is your WBC. This is your bacteria. So your WBC have to consume this bacteria, which is opsonized. So your bacteria is coated with certain tasty substance. So how does this happen? Your WBC will have certain receptor. It will have certain receptor which can go and bind to those opsonized things so that it can go and bind. Okay, so you have something called, it has to recognize these opsonins and attach to them. That is recognizing an attachment by means of receptors. Next, after recognizing, your WBC have to engulf. It have to engulf like this, the bacterial product. It has to engulf your bacteria. So how does it engulf and what does it do after engulfing? Now, successfully, your WBC has engulfed the bacteria, and inside the WBCs, you will have a lot of organelles. One organelle is called lysosome. So what is lysosome? It's a membrane-bound organelle. Like you have your nucleus, you have other organelles like lysosomes. This contains a lot of enzymes, especially acidic enzymes, which when released can kill the bacteria. Okay, so it forms a vacuole or it forms a a bubble thing around the bacteria, and your WBC will form something called phagosome. So this phagosome has to fuse with lysosome so that the enzymes are released, your bacteria is killed. So this is called phagolysosome fusion. Phagolysosome fusion. So only when this fusion happens, the acidic enzymes will kill the bacteria. So let's imagine there is some problem with this phagolysosome fusion. What will happen? Your bacteria will not be killed. And these lysosomes inside the WBCs, they will appear like big granules because of defective fusion. So when your phagosome is not going to fuse with lysosome, you have a disease called Shidak-Higashi syndrome, one of the characteristic feature which you get to see in peripheral smear is giant granules, especially in the neutrophils. And this is due to defective phagolysosome fusion. So, associated with this Chidak-Higashi, you can have albinism also. So, why do you have albinism? That's because you have something called melanosome. Your melanosome also have to fuse and release all your melanin. So because there is some basic defect in this fusion, your melanin also will not be properly reaching your skin or synthesized. So it will be synthesized, but you have some problem with melanin, you get albinism also. So Chidak-Higashi can be associated with giant granules and albinism. And apart from that, you have so many yans, which is seen in Chidak-Higashi. You can see nerve defect. The patient can have nerve defects. They can have nystagmus. Okay, they can have nystagmus, and their natural killer cells functioning can also be affected. So all these can occur in case of your Chidak-Higashi syndrome. And that's due to defective phagolysosome fusion. So we are done with absonization, coating and everything. The next final step is phagocytosis and killing. It's all in the process of phagocytosis. Now you have recognized and engulfed. Now you have to release certain enzymes and kill those microbes. So this is done by a powerful mechanism called the oxygen halide system. It's called the oxygen halide system. So I want all of you to listen to this very carefully, how your microbe is being killed. Okay. So first your oxygen will be converted into a superoxide. Your superoxide will be converted to hydrogen peroxide, and hydrogen peroxide will be converted to hypochlorous acid, which is a powerful microbicidal agent. And for each of these steps, you require certain enzymes. So the enzyme which helps in conversion of oxygen to superoxide is NADPH oxidase. Whereas from superoxide to hydrogen peroxide, you have superoxide dismutase. Whereas from hydrogen peroxide to hypochlorous acid, you have an enzyme called myeloperoxidase. So if you have deficiency of any of these, again, you can have some diseases. Like I told, if you have deficiency of integrin, you can get LAD1. So like that, if you have deficiency of NADPH oxidase, you can get a disease called chronic granulomatous disease. It's called chronic granulomatous disease, right? So what happens in that disease, I'll tell you. Because if you don't have NADPH oxidase, you don't have your hypochlorous acid generated, so the organism will not be killed. So these patients will have recurrent granulomatous inflammation in any part of the body, especially your genitourinary tract or your GIT. Okay. Likewise, if you don't have myeloperoxidase enzyme, again, you will have recurrent infection, and this disease is called MPO deficiency. And similarly, if you don't have the superoxide dismutase, you can have a disease, which I'll show with a picture later. So now, let me give you a task, and if this concept is clear, you will be able to understand the task which I'm going to give you right now. So basically, your NADPH oxidase is going to generate superoxide, which will be converted into hypochlorous acid by MPO. So you have two conditions. One is chronic granulomatous disease due to deficiency of NADPH oxidase, and MPO deficiency, which is due to deficiency of myeloperoxidase. Now let me give you a task. You have two patients, patient 1 and patient 2, and you expose both the patients to catalase positive and catalase negative organism. So I hope those who have read microbiology, you know what is a catalase. So certain microorganisms can produce an enzyme called catalase. Those which do not produce are called catalase negative. So example for catalase positive is Staphylococcus. Catalase negative organism is Streptococcus. So you have two patients who have some enzyme deficiency, of which I've told previously. You expose both the patient to catalase positive and catalase negative organism. So this patient number 1 is getting infection only by catalase positive organism, whereas this patient is able to kill the catalase negative organism. But patient number 2 is getting infected by both the organism. So can you guess why? So you have two patients. One patient is getting infection only by catalase positive, whereas the other patient is getting infected by both catalase positive and negative organism. Why is that happening? So that's again, if you apply here, so patient number 1 has got chronic granulomatous disease. So I told these patients get infected only by catalase positive organisms. Why? Because if you have a catalase negative organism, some catalase negative organism can produce their own hydrogen peroxide. So even if you have deficiency of NADPH oxidase, the negative organism can produce their own hydrogen peroxide, which can lead to produce hypochlorous acid, which can kill. Okay. But if you have MPO deficiency, what happens? Even if your catalase negative is going to produce hydrogen peroxide, it cannot yield the end product hypochlorous acid. So MPO deficiency will have infection by both positive and negative organism. I hope this point is clear. So remember, patient number 1 has got chronic granulomatous disease. Patient number 2 has got MPO deficiency. So if you have any doubt with this and why this occurs, just look at this one. That's because your negative organisms can produce your hydrogen peroxide. So that can get converted into hypochlorous acid. So you can rewind and watch this concept to understand what happens in these two conditions. Okay. So remember, chronic granulomatous disease is due to NADPH oxidase deficiency. So now we are almost at the end of discussion. So let me summarize whatever I've told so far. So I've told about margination, rolling, adhesion, transmigration, chemotaxis, opsonization, then phagocytosis and killing. So far I've been like a lengthy lecture very slowly. So rolling is because of selectants, adhesion is because of integrins, transmigration is because of CD31 or PCAM, chemotaxis is because of CIL, your complement 5A, interleukin 8 and leukotriene B4. Your opsonization is because of CSF, your complement 3B, then your serum proteins, FC fragment of immunoglobulins, your serum proteins and FC fragment of immunoglobulin. And this is because of all your NADPH oxidase and your myeloperoxidase. So as I told in the beginning, if you have defect, you have some associated disease with it. So if you have defect in selectins, you have LAD2. If you have defect in integrin, you have LAD1, which is associated with delayed separation of umbilical cord. If you have defective opsonization, your immunoglobulins are not there. You have a disease called Bruton's A gamma globulinemia because this is absence of gamma globulin. So because you don't have these immunoglobulin, your opsonization will not happen. So Bruton's A gamma globulinemia is a defective opsonization. You'll be learning in immunity. Okay. And if you have NADPH oxidase deficiency, you get chronic granulomatous disease. If you have MPO deficiency, you have the disease is called MPO deficiency. And chronic granulomatous disease, you have infection by catalase positive organism, whereas MPO, you have infection by both positive and negative organisms. So you can just again watch this video and then come back to the MCQs. So we'll move on to some MCQs, which is based on the cellular events of inflammation. So I hope most of you will be able to answer this. So given below is a cellular event of inflammation. One event is highlighted by green, that is this particular event. So which of the following is responsible for this? So what is this particular event depicted here? Anyone? So it's just coming out of a blood vessel. This is called diapedesis or transmigration. So what's the answer here? So for transmigration, the most important one is CD31 or PCAM1. So the answer is CD31 or PCAM1. Okay. So this must be the answer. So next question, A 5-month-old child was brought to pediatric OPD with recurrent skin infections, and there is no family history of similar complaints. There is delayed separation of umbilical cord. So, where did I mention about this delayed separation? Does anyone remember? I told your leukocyte has to adhere your umbilical the umbilical cord will be released. I mean, it will be falling off. So, delayed separation of umbilical cord is seen in leukocyte adhesion deficiency 1. So, remember 1 and I, integrin defect. So, the given condition is LAD1 and the answer is integrin. It's due to defect in integrins. So, next question. So, whoever has answered this has listened very carefully. So it's very typical. A child with recurrent infection, the appearance is shown here and the peripheral smear is shown here. What's your diagnosis? So it has got here, the child has got a white skin and a white hair. This is albinism. And what cell is this? As I told in the first beginning session, it has got multiple lobes. So it's a neutrophil, and you're seeing these giant granules. So where did I tell about giant granules and albinism? So there was one process, your phagolysosome fusion. If this doesn't happen, you get a disease called Chidak-Higashi syndrome. So this particular disease is called Chidak-Higashi syndrome, where you get giant granules and albinism. Whereas, where do you get chronic granulomatous disease? It's due to NADPH oxidase deficiency. Okay. So, SID is severe combined immunodeficiency syndrome, which we'll be discussing in immunity. That's due to adenosine deaminase deficiency. Adenosine deaminase and other enzyme deficiencies as well. So, that will be discussed in immunity, right? So, now, can you guess who is this and what disease this person has got? So, this is Stephen Hawkins. What disease did he have? Anyone? He had amyotrophic lateral sclerosis. He got amyotrophic lateral sclerosis. So, what disease is this to do with all these events? So, I don't know how many of you have noticed when I was discussing about these NADPH oxidase and other stuff. Let me go back to that. Okay, so I told you about an enzyme called superoxide dismutase. So when you don't have this enzyme superoxide dismutase, which is an antioxidant, you get amyotrophic lateral sclerosis. So this is how your inflammation is related to all your clinical conditions. It's related to all your clinical conditions. So whenever you see a patient, you go back, it's always correlating with pathology. So next question, a PhD research fellow in hematology plans his thesis on the mechanism of inflammation. He observes that the leukocytes move from the blood vessel to the site of the microbe, which is likely to mediate this. So, are you able to understand the question? So, he is going to study something on how your WBCs are moving to the site of the microbe. Let me consider this is the bacteria. So, what is this particular event called as? It is called chemotaxis. So, in other words, this question is about the chemotactic agent. So, what is the mnemonic which I told? It is CIL, complement 5A, interleukin 8, and leukotriene B4. So, what's the answer here? It is complement 5A. What about the other options? These are opsonins, which coat the bacteria and makes them tasty. Not bradykinin, but option A and C. So, option A and C is going to act as opsonizing agents. So I have put up each of the question so that it will be easy for you. So we'll discuss at some other video sometime about what will happen to your systemic changes in inflammation. I thought I'll discuss in this video, but it will be too much of time consuming and overloading. So today what we have discussed is about what is inflammation, what is acute and chronic inflammation, what are the signs of inflammation, what are the vascular events, what are the cellular events, and in cellular events we have seen, of course, for each of the events, so for margination, rolling, addition, for everything we have discussed about what is the associated disease and all those. Okay, so always pathology is about correlating. So don't think it will go off with your second year. So it's going to come with you for lifelong. So whatever medicine case or surgery case you see, if you open the pathology book, you're definitely going to find something. And that's how the treatment and everything goes on based on the diagnosis. So for those who are in second year, my suggestion would be try to read Robbins. I know many of you follow a lot of books, so try to at least read Robbins. You're not going to sit and read Robbins there, it's going to be tedious. So try to read at least Robbins once when you're in your second year or whenever you get time. Okay, it's very interesting to read. So thank you all for watching this video, and thanks to Medicine for this opportunity. So you can ping me at Facebook or YouTube. We have a channel called Pathopulse_Radhika where I post all YouTube videos, short YouTube videos based on clinical scenarios. So I'm very happy to take any questions, if any. And always the key to success is to focus on the goals, not on the obstacles. Okay, so keep going and wish you good luck. Thank you so much. Any questions? If any questions, I'll be happy to take any questions. Yeah, Raghu, is there any questions to take up?
Hope there is no questions for now, ma'am. If there is any question in future, I guess it will be is good if you answer them in the session, ma'am. In the video.
Yeah, definitely. Sure.
Thank you so much, ma'am. Thank you so much. It
I
was
can
a
see
great
something.
session.
Okay.
Yes, ma'am.
Thank you.
And it was a
No,
great
I was
session,
just seeing the comments like answer the questions.
Yeah, ma'am. It was a eye-opening session really to understand how to correlate and read clinically. If we learn in this way, it will be really good to prepare for next or INICET or any.
I think your voice is breaking.
Pleasure in having you in the session, ma'am.
Thank you so much.